Okay, I'm back. Let me see if I'm totally back or if I still have a problem like last time. It wasn't allowing me to share my screen. There we are. There we are. Okay, so let me bring chat up just in case. Chat. Okay, just organizing my screen here. Okay, is everybody ready to go? Let's go. We're on slide 16. Sydney's ready. Thank God. Thank God, Sydney. Thank you. Okay, slide 16. So in last class, we talked about adherens junctions and integrin junctions. So adherens make, what are they called? Cadherins make adherens junctions. They also make desmosomes. And your integrins are mostly focal adhesions. So we're going to be talking about those two different things today and how they're used. Basically, adherens junctions are for holding close tightly to other cells in tissues. Focal adhesions can be used to stay in a tissue by holding on to the extracellular matrix, or can be used to crawl along extracellular matrix and move. So we'll talk about both examples today. Here on slide 16, it shows you how cells can rearrange themselves based on their adherens junction. So remember, adherens junctions are always same. So you have to have the same cadherin in order to join to a cell. So here we've got two different types of cells. We've got mesodermal cells and ectodermal cells. So mesoderm is going to stick to mesoderm, ectoderm is going to stick to ectoderm, yes. So what they did was take these two different tissues, disperse them into individual cells, put them back together and find out what happens. What happens is the mesodermal cells kind of sort themselves until they end up touching each other and locking in, and the ectodermal cells do the same. So they automatically resort. And part of that is because of the selective adhering between the like cells. And part of it is because when like cells adhere, they get the surface tension. So they like grab, and then you get actin myosin in the cortex that provides some tension. And that, don't ask me how, causes them to move a little bit. So they're moving a little bit and sticking where they stick, and then they automatically sort. Kind of makes me think of, although I didn't read this, but I'd be willing to bet, if you go back to early mouse development, we talked about how you've got some cells that express OCT4 and some are expressing CDX2, and they're separate. But the OCT4 cells, randomly, some start making more NANOG and some make more GATA4, and they're just kind of interspersed. And then by magic, it seems, based on the book, because the book doesn't tell us, by magic, they automatically sort. So all the OCT4 cells NANOG cells are together, and the GATA6 cells are together. So I wouldn't be surprised if it had something to do with this, something like this going on, where you've got expression of cadherins that cause the cells to resort. So anyway, as a general mechanism, that's what's going on here. You've got cadherins that only attach to other cadherins, and so the cells sort themselves. Exit or slide 17. Okay, so we've talked a bit about these transitions from endothelial to mesenchymal cells, and from mesenchymal cells to endothelial cells. We've talked about this before a number of times. Hang on, I'm just trying to pull my notes up here so I can remember where we were. Okay, yeah. So this is kind of important because in embryo development, epithelial cells, for one thing, can be induced to become a lot of different things. So a lot of things start out as epithelial cells that eventually become other tissues. And also, these endothelial cells can because they stick together with E-cadherin, they can be used to model. So they're, you put stress on it and it will stay together and then form. So a lot of things happen because of epithelial cells that are hanging onto each other and then being formed into different structures like tubes, the neural tube and the blood vessels and things like that. And other kind of folding goes on because of these epithelial cells. So epithelial cells are really important. Normally in adult cells, we think of epithelial cells as skin, skin cells on top of on your surface, also endothelial cells, which are cells inside. And if you're in my cell biology class right now, or if you've taken before, I talked about how inside that epithelial cell layer, there is a layer of there's a basal lamina, really thick protein coat between epithelial cells and the inside of the body. And that's important for protection of the body from external invaders. Here in the embryo, though, we don't have that distinction between epithelial cells and inside cells with the basal lamina separating them. Epithelial cells are more generalized. They're skin that can be molded and then turned into something else. So I hope that wasn't too confusing, but that's the way I think about it. Okay, mesenchymal cells then are loosely connected. We've talked about these several times. In fact, we talked about this early on when I had no idea what a mesenchymal cell was. Do you remember that? I said, this is what it means. And you're like, is that what it means? Are you sure, Dr. Slish? Oh, yeah, this is what it means. And I looked it up and found out what it meant. And what it actually means are these cells that are loosely connected, if they're connected at all, and they crawl around the cells. I mean, crawl around the embryo and go somewhere else. You have mesenchymal cells in the adult, in that dermal layer, I believe. If you remember a couple lectures ago, I talked about the neural crest and the somites. Part of the somites became dermis, and then you've got epidermis on top. So in the adult, you've got epidermis on top, basal lamina, and then the dermis. You've got mesenchymal cells that can crawl through that dermis and fix any damage, like fibroblasts will go in there and release collagen to scar it up, so to fix problems. So that's in adult cells. In our embryos, we've talked about them before, neural crest cells crawling around, cells coming from the stem zone, crawling through the node, and then going anterior and lateral to that. Those are mesenchymal cells. So we've talked about them quite a bit already. They may be connected to each other by gap junctions. So we haven't talked about gap junctions yet. I keep thinking everyone should have cell biology before this, because all this crap I talk about in cell biology. But anyway, if you look at that little picture on the bottom of slide 17, those are some pretty schematic looks at gap junctions. So they're basically a channel in one cell and a channel in another cell, and they connect to each other. Now there's a channel from one cell to the other. And they're regulated, so it's not like free flow of anything, but a lot of things can go through there, small molecules mostly. I don't think proteins can go through, but any small molecule will go through. I talk about these a lot in cell biology. They connect cells, like cells in the heart are electrically connected through gap junctions. They allow ions to go through, and one depolarized cell depolarizes another cell. Here you can see, I don't know what's passing back and forth here in these embryonic cells, but you can see in the third slide, or third part of the upper figure here, you can see they're barely touching these mesenchymal cells. And that's through gap junctions. So what this slide is trying to show is you've got these red solid junctions between these epithelial cells. And here you do have a basement membrane, which is your basal lamina. But these cells then let go of those cadherin junctions. And when they do, they crawl off. And again, if you're in cell biology, we talked about this in the last class of cell biology. This is part of the transition of cells from normal cells to cancer or malignant cells, because they let go of the cells next to them. They start crawling and moving in a certain direction and invading other tissues. So that's another example of a epithelial to mesenchymal transition. So you can see again back to cell biology. that's the cells are becoming more embryonic, so they're becoming like this tissue type in embryo where the cells can move around a lot. Okay. Okay, Dr. Slush. Keep talking, keep talking, keep talking. Okay, so that's about it. All I wanted to say about that epithelial to mesenchymal transition is about how, again, it's regulation of those cadherins. And when the cadherins let go, then the cells let go. Is this all referring to the mouse? This is general. This is just, this is general to all things. I think later we'll talk more specifically about the mouse and the xenopus for sure. Oh, actually, right now we're going to talk about the mouse. But when I talk about the stuff I've talked about so far is all general, happens in all embryos. Okay, compaction of the morula. Now we're talking mammals, specifically mouse. So let's go on to slide 18. 18. Hello, 18. Okay, so here you can see a couple different versions of that morula. In the top one, it's scanning electron microscopy, which is when you put something opaque on the outside of your cells and you shoot electrons off of it. You get a really nice picture from it. So here on the top, you're going from 8 cells of a normal embryo into compacted 8 cells. It looks a lot more clear in the light or more obvious that you're compacting in the light microscope. Because here on the left, you've got 8 separate cells, and on the right, it looks like one big glob of cell. So that's what compaction's all about here. So in compaction, your epithelial cells lose their adherens junctions. Oops. Epithelial cells lose their adherens junction. Oh, what? No. Oh, I never did this. Oh, shoot. Yeah, this is, okay, here's, looking at slide 18, you can see how I make my slides. Basically, to make sure everything's the same color, I duplicate the last slide and change the words. I didn't change the words here, so let's annotate this slide as long as we're here. Compaction. Early mammalian embryos increased surface contact through adherens junctions. Okay, there we are. You just had a peek behind the curtain. That's the way it's done. Okay. Wait. I'm sorry. You know, I'm going to update this PowerPoint and hand it up, and then hand it out later because yeah, I never finished that slide. But anyway, what's going on here in your 8 cell? If you remember back to that, I think it's chapter 5. We talked about this. Yes, first half of lecture 5. So you've got 8 cells. And they're they're connected, but they're not tightly connected. And then they start expressing their adherens junctions, and then they start to compact. And they're compacting because some cells, like the ones on the inside, are already connected on all sides. And every time you have a connection, like I talked about a little bit earlier, those connections cause tension to happen in that the cortex of the cell. So these adherens junctions form and then they contract and you get surface tension in the cell. Now, if you've got a, if the cell is connected on all sides, you've got surface tension on all sides, and that makes it one type of cell, basically. If it's on the outside and it's got an apical surface that isn't connected to anything, it's got less surface tension because the surface tension only happens where there's connection. So what happens here is, and the kind of the segregation here that happens early in this morula when it goes to the 16 cell stage is if you've got a lot of surface tension, high surface tension, these cells move inside. and lower surface tension, the ones that already have a free side, go to the outside. And that then causes the activation of the hippo pathway on the inside of the cell, again, chapter 5, first half. And that hippo pathway will downregulate CDX2 and upregulate OCT4. So on the inside, you get OCT4, and the outside, you get CDX2, and that's your trophectoderm versus, the embryo proper there. So, last time we just said it happens, and now we see why it happens. It has to do with surface tension, which is occurring because of the free apical surface, although we did kind of say that last time too. Okay, everybody happy with that? Morila, let me just sum that up because I was talking a lot there, not saying much. So during that 16-cell development, you've got cleavages that happen. If you read the textbook, they get a little complicated about how those cleavages happen. I'm skipping over that. But after the cleavage to 16 cell, the cells that have connections everywhere, have more surface tension, they segregate to the middle of the embryo. The ones that have some apical surface will, migrate out. And then you've got CDX2 on the outside, OCT4 on the inside because of activation of that hippo pathway on the inside ones that have a lot of surface tension. Good? I was just blowing dog hair off. Sorry about that. Okay, Xenopus gastrulation. Gastrulation. Okay. Slide 19. So here we're going all the way back to the first week of class in gastrulation and Xenopus embryo. And again, this is kind of the chapter where before we said this happens, and now we're going to see why it happens or more in depth about why these things are happening. So here with in gastrulation, we've got two different cell movements that are important. One is the involution of mesoderm and endodermal cells, and they're crawling across the ectodermal cells here. That's your actual gastrulation. And the other thing that's happening during gastrulation is the epiboly, or epiboly, I guess it's pronounced. And that's when the ectodermal cells, which are animal cap, spread all over the whole thing. So you end up with skin all over your body instead of just on part of your body, at least if you're a frog. So why is this happening? Well, the interesting thing about this is it has to do, it isn't because, I think even in chapter 3, I said cells are dividing and you're getting more cells. Not true, it turns out. I was lying. Nobody caught me though. Okay, so actually what's happening is something called convergence and extension. And it happens with the epiboly, but it's more important with the gastrulation movements because convergence and extension, I think the best way to think about this is kind of like when you've got some clay, you've got like this round cylinder of clay, you put it on the table, you roll it like this, and when you do, it spreads out. And so you've got the same clay molecules, but now instead of being in a little cylinder, they're in a long tube. Another way of looking at this is extrusion. So if you look, has anyone played with Play-Doh? Any of you? Can I get a thumbs up, thumbs down? No one? No one knows what Play-Doh is? Thank you, Adya. Thank you. Okay, Play-Doh. This is extrusion, or I don't think anyone has. Thank you, Adelina. So you get a little chamber here, you put a big glob of Play-Doh in there, you crank down on it, and it gets squeezed out a little tube. Well, the same thing's happening here with our embryo, in that the hole is the blastopore. And now you've got all these cells of mesoderm that are all the way around the embryo, and they're all coming and squeezing through that hole. So when they do, they get drawn out. And if you look at the gastrulation in like time-lapse video, you can see tons of these. Xenopus time-lapse video gastrulation. And the thing goes from a round ball to a stretched out embryo, and now you've got head, tail, left, right, blah, blah, blah. So these things are coming in through the pore and squeezing up through. So how does that happen? you're not getting cells going through because they're dividing. There's no cell division here. They're actually moving through that hole. And as they're moving through that hole, let me just pull up my notes here real quick so I don't say something wrong. So, so there's two different mechanisms of convergence and extension. One is where the cells, say you've got a flat sheet of cells, and they kind of, if you're extending in this direction, let's look at the figure. Figure 22. So here you've got a flat sheet of cells, and their long axis is kind of perpendicular to the axis they're going to extend on. And they kind of go, well, they start out like this, and they start out like this, and they go, So you go from flat to long, and you can see them kind of mixing amongst each other here. Okay, so you've got the middle ones and these, and it's called intercalation. They go across amongst each other, basically. And that's going to extend and narrow that tissue. And the other is mesenchymal crawling, which is basically the cells just move towards one area, and that's convergence, and they extend when they get there. So I believe we talked about that with early chick embryo, the formation of the primitive streak is that kind of convergence and extension. I'm not 100% on that, so don't quote me, but if I ask you about it on the test, I'll just mark it wrong, whatever you put, so who cares? C&E causes so this is cool. When you finally get kind of this three-dimensional understanding of what's going on, your convergence and extension is causing the equatorial band of mesoderm to squeeze through the blastopore. And when it does, it becomes a long sheet of mesoderm layered between ectoderm and endoderm. This is like biological magic. So if you look at slide 23, this is the top one is you're just looking at the mesoderm from the xenopus embryo. And then over at the dorsal side is where it's all going to get squeezed in and through. So you can see how everything gets pulled up to that spot and then squeezed and flattened out. So that's your convergence and extension. And it's happening because all these cells in mesoderm are connected to each other. So when these move, the other ones get pulled with it. And as they hit that focus point, the blastopore, they intercalate with each other and become long. So that's converting that equatorial band into a sheet in the trilaminar gastrula. Don't you love it when I throw out words like trilaminar? I hope you know what that means. And then the interesting thing about that too is, if you look here, they've labeled some of this stuff. So you've got B and A here, C and D here, and I think in the next one they've labeled some other parts. But look how C and D are on opposite sides of the embryo, right? And A and B are right next to each other. B is going to be kind of on top of A, and A is going to be going in through the blastopore, but B is staying where it is. So let's look at 24. So here's B and A on top of each other. Now A is going down and in and across the roof there of the blastopore. And when it does that, A ends up at the leading edge of that gastrulation, so it ends up anterior. B stayed where it was, so it's posterior. And so you had this A and B that were right next to each other, and as it moves, now they're apart from each other. But at the same time, you've got the sides of the embryo coming in and going through. So now you've got things that were on the opposite sides of the embryo next to each other. So if you look at C and D, over here, they're on opposite sides of the embryo. And then after neurulation, right, you see your somites here, they're right next to each other on opposite sides of the embryo. On opposite sides of the neural tube, sorry. Convergence and extension. In moving cells. Cells are polarized in the direction of the animal cells. So how is this happening? How is this gastrulation happening? Well, you've got cells, you've got your mesodermal cells are connected to each other, and they don't have cadherin connections to the ectodermal cells. If they did, they'd be stuck, right? But they do have adherent junctions, or no, I think they're desmosomes, because they're connections between different cadherins, because different tissues have different cadherins. So you've got your mesodermal cadherins and your endodermal cadherins, and they're grabbing each other in these hetero whatever desmosomes, and they're connected to. Bottom line, they're connected to each other. So the mesoderm and endoderm are connected to each other, but they're not connected to the ectoderm. But you still have extracellular matrix there. So inside that blastopore, the blastocele, you've got ectoderm across the top, and underneath that ectoderm, there is extracellular matrix, proteins, right? Fibronectin being one of them, collagen and fibronectin, right? So as these cells go in, they're doing the cell crawling thing, where they're reaching out and grabbing with their focal adhesions and pulling the cells forward, reaching out, grabbing. So they're crawling across the extracellular matrix of that ectoderm by cell crawling. And they can move with respect to the ectoderm because there's no adherens junctions there. After they totally stretch out, then they'll form those adherens junctions and they'll be stuck. But so what we've got going on here is you've got polarized fibrils of fibronectin in the extracellular matrix of the blastocele roof. I love that term, blastocele roof. So it's a blastocele, and it's like the top of it, which is the underside of the ectoderm. And they are polarized. So that polarizes the movement of the cells, so they're not just going to come in and go anywhere. They go straight to the anterior end. There's also an increase in the surface tension in the epithelial cells of the blastocele roof. So these ectodermal cells have got adherens junction between each other. And again, whenever you get those adherens junctions, you get surface tension. And that's going to, that surface tension then gets transmitted to the extracellular matrix through integrins in focal adhesions. So think about this. In those ectodermal cells, you've got adherens junctions between them, right? Adherens junctions are cadherin to cadherin connections that connect to their cytoskeleton. So they're grabbing each other, and you've got actin and myosin that's increasing surface tension. That increased surface tension is related to the cytoskeleton. Cytoskeleton is attached to the focal adhesions. Focal adhesions are attached to the extracellular matrix. So because of these adherens junctions doing this, the integrins are grabbing that extracellular matrix, and that's putting surface tension on the extracellular matrix. And then your mesodermal cells are being, I guess, stimulated by that surface tension, and that's helping them to crawl across the top. It's beautiful stuff, people. Okay, so I guess that's it. I guess I've said my piece. Now I'm going home. Oh, I am home. Chat. All right, any questions about that? I think I did it fast, but I'm not sure. Any questions on that gastrulation, that aspect of gastrulation? Nobody? Nope. 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 Good Lord. Okay. All right, so the last thing to talk about today then is, hang on. Got to rearrange my screen. Oh. Okay, so the last thing to talk about then, and I don't really have a lot of slides on this, maybe I'll throw some more figures in there, but the epibole. the epilaceous movements. I just made that word up. Okay, so epibole then is that ectoderm spreading over. Now here again is the interesting thing that I think I said incorrectly back at the beginning of the class. I think at the beginning of the class I said you've got cell divisions that are causing that, which is kind of true in that you're getting more and fewer cells, but you also have like a thicker layer of ectoderm. So it's not like a single layer of cells. You've got several layers of cells in that animal cap. And part of that epibole, or all of the epibole, is being caused by not cells dividing, but cells intercalating and spreading. So you've got like, 3, 4 layers thick, and they thin down to one layer. And as they do that, they're spreading out. So here we're talking about taking a ball of dough and a rolling pin and rolling it out. Got the same amount of flour, but now it's flat, and so it's got a larger surface area. So this thick layer of ectoderm is thinning out and spreading, going around the cell. And that's radial intercalation rather than linear intercalation. I think that makes sense. Yeah. Okay. Preguntas? Any questions? I think that's about all I have prepared for today. I'm going to mute Christina because I'm tired of listening to her. Boom, you're mute. Just kidding. I can unmute you from here. No, I can't. Shoot. Sometimes I try to sneak up on people that way, but it doesn't necessarily work. Okay, you got no questions? Can I get a thumbs up from people? Let me know you're alive. Boom, ba-boom, boom, ba-ba-ba-boom, boom, ba-ba-boom. Michaela, you're not thumbs upping me. Michelle Bellavamento. 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 Hi.
Hi, can you answer Sydney's question in the chat?
Can I ask what?
Can you answer Sydney's question in the chat? I didn't hear if you answered it.
Whose question?
Sydney, she has a question in the chat.
Oh, Sydney Adolfo. Silva says we have an exam 4 on Wednesday. No, we're not, because we're not having a final. I think I said this before, but you weren't around. No, we're having one more lecture on Wednesday, which may involve some review depending on how much new notes I get. And then we'll only have one more exam, this exam 4, which will be in finals week next week.
Thank you.
'13th, I believe. You got to believe in something. I just hope organic chemistry doesn't get in the way. I think organic chemistry has been screwing up my cell biology class lately. Oh, they moved that test. Oh, good. So that would be this Thursday, 7 a.m. Oh, they don't care about you. So basically, it would be better for you to drink coffee at midnight and stay up till 7, because you're not getting up at 6. Let's be honest. So Nick, I don't know. Look that up in Banner. I haven't looked at it yet. I'm not sure what time it is. Wednesday at 2:30. Everybody's got that. Okay. So any questions, or should we call it a day? Calling it a day. There's these primates. I don't know, some kind of monkey, I think, and they threaten each other like this. They flash their eyebrows. May the fourth be with you, and also with you. Sydney's calling it a day. Let's all call it a day. Okay. I Hope to see you again here Wednesday. Take it easy, dudes.